Hi everyone. I will admit I'm a bit nervous presenting at a biometrics conference. I'm only a baby in biometrics, but I've been guaranteed that it's the nicest conference on the planet. But I'm really excited to talk to you all about two of my favorite things, the first being chickpea and the second being Australia. So all of you should know what chickpea is, I hope, or have eaten it at some point. But what's really interesting about chickpea breeding in Australia is that it's operated by a small public breeding program. That's Christy there in the picture. She's incredible, but she's breeding for a really broad, diverse geographical landscape. So chickpea is produced primarily in the northern region here. Okay, it comes up as sometimes the white disappears and it looks like islands, but it also is produced in the southern region and is now expanding into the west. And so the way the program has been making decisions is by splitting up these three regions into two, so the north and the south. And so for those who are familiar with Australia, it is ginormous. And for those who are not, I've slapped it on Europe, and you can see that even the entirety of the UK is smaller than the northern region. So you can imagine that trying to make decisions for such a broad landscape is quite challenging. And when we think, my ecology lecturer once said to me that Australia is a mosaic of ecotypes. And so what that means is not only across these really broad ranges, but even in small regions within the northern location, you get really diverse soil types, weather conditions, but you also get really diverse extreme weather events and the frequency of those weather events. And I've put a few pictures here of just some of those extreme weather events that we've had just in the last 12 months. But to be honest, the last 3 years have been a nightmare. And so it really raises the question of how do we make targeted decisions? And also there's a growing interest in how can we breed for climate resilience. And so the project I'm actually employed under is looking at heat specifically, but obviously it's a complex interactions of a range of covariates. And so because of that, there's a real question in my project was, we have all this information, historical breeding data. Could we potentially leverage that data and disentangle stress adaptation for selection? And so in order to do that, there was really two key questions. Can we better characterize the TPE that chickpea is currently grown in to understand how G/E is structured both temporally and spatially, so over time and across regions, and what covariates are driving the composition of the TPE? So I'm really lucky that I get to work closely with the breeding program, and they have a really great resource of data. So this is just kind of some of the locations that they breed for, split into that north and south that I mentioned, but I'll just be focusing on the south today. But just within that southern region, it contains 26 locations, 63 environments, nearly 5,000 unique genotypes, which results in over 300,000 G/E combinations. And to really strengthen the analysis, I've used contemporary groups, which essentially just groups different stages within a year in an environment together. And that allows us to really improve concurrence across years, as well as allows us to reduce potential selection bias for later stage material. And also having the early and later material means that we can tap into potentially earlier material that may have been thrown out due to it not being very tolerant to disease, but may actually be quite resilient to particular stresses. And so using this data, I ran a regression type model. So there's a range that you can use. I obviously use a factor analytic model, which is really great at modeling G/E, and it's really great at capturing the complex G/E interactions across environments. And so a lot of people obviously use this model and make a lot of selection decisions based on it, but I guess how could we add value to those outputs and kind of tease apart this G/E more to get more information about, more in particular, stress? And so I might not be a biometrician, but I sure know a few smart ones. And I'm really lucky that I get to work with Daniel and use a new tool that he's developed called GE Tools, which isn't available on GitHub, but is being rolled out, so just send them an email. But essentially what this tool does, it allows us to take the outputs from these regression type models. So in my case, I took the variance covariance matrix from the FA model. It applies a rotation, which allows us to transform this G/E component into two independent components, the first being non-crossover, which is aligned to the genetic effect mean or the average across environments. And I think a few people have spoken a lot today about scale and non-scale and non-crossover and re-ranking. So that non-crossover really aligns to that scale G/E. And the second component is the crossover, so how genotypes re-rank across environments. And so this tool is really great because it's built within selection index theory, and I'll show some examples of that later on, but it just really aligns how non-crossover G by E can be aligned with overall TPE, so with all the environments within the southern region, as well as within a single location. So a bit about the data. So I ran the FA model, and this plot to the left is showing the heat map. So each individual block is an environment and the correlation between the two environments. And so you can see here that there is some heterogeneity in the data. So there's some environments that are really strongly correlated, and there's others that have a moderate to weak and even negative. And that's represented also by the plot to the right, which is just showing the distribution of those correlations. And so we can kind of take a look at this output further by grouping them by locations. So in my case, I've just grouped them by states. And you can see that there is a bit of structure, but kind of not as much. And when you look at the correlations between states and within states, the mean is the same, which kind of indicates that it's not as, state isn't a major structure. And then I guess when you have a look at the next slide, which is looking at year, and you can see that the mean correlation within a year is much higher than between, you can see that there is definitely a year effect. And that's really challenging to how do you breed for new years, especially when they keep saying we have 1 in 100-year weather events, but it's happened for the last 3 years where I've been growing field trials. So, but yeah, how can we take this data and kind of pull it apart even further? And so here we have, on the left, the bar charts. I've taken 2 locations, Horsham, which is this red point here in New South Wales, and then I've also taken Scaddan, which is in Western Australia, and they have multiple years across 2018 to 2023, and to the left I've just got each year, and each colour represents the proportion of crossover and non-crossover G by E. And so the orange, for those who can't see the label, sorry, that is non-crossover, and then the blue is crossover. And you can see already like how temporally G by E and these 2 components change over time, and you can see how different they are across different seasons. So making decisions about which sites are more stable than others, and thinking about that composition is really important. And you can see how the crossover and non-crossover also, as you would imagine, influences genotype performance and that re-ranking. And often you would see these plots and people say there's crossover and non-crossover, but the great thing about this tool is now you can actually quantify it. And so using this information, we can then look at how these environments are aligning to the overall TPE and also within a location. So these plots here are for Horsham and Scaddan, and on the left is the alignment to the overall TPE. So each year, it's how well correlated it is to the overall TPE for all of the environments within the southern region, and then alignment to the site. I probably should have put location, but that's if you were to just take the subset of the locations and just look within the location. So the first key feature of these plots I will show is all of these sites here that have a high correlation with the overall TPE and the site-specific TPE. We could consider them quite reliable sites for driving genetic gain. But what's interesting here is you can also see that there's some sites in red here that have poor alignment to both overall TPE and the site. And so I guess to me is why are they so misaligned? And potentially could they be probing environments? Because obviously these sites are linked to high crossover G by E. Are they potentially sites that are experiencing stress? So I guess the big thing about historical breeding data, you don't get a list that say this site's been hot, this site's been dry. So we need to try and disentangle some of that information from this large data set. I also wanted to flag in orange these 2 sites that have a high alignment to the site, but they have a low alignment to the overall TPE. And so using this information, can we kind of use it as, I mean, people can use it for selection decisions, but it does a diagnostic to try and identify stress-prone environments. And if these environments are linked to particular stresses, can we then, when we're making selections, weight these environments more to start for breeding for material that may be more adaptive or resilient to these stresses? So I wanted to use this information and link it to some environmental covariates. So I've just taken raw covariates that we get from a weather station. I've also used some of them to calculate more biologically meaningful covariates, like growing degree days and photothermal quotient adjusted with VPD. And essentially I got daily values across the entire season, and I just summed them up to just get one value for across the season, which I know people have done and it's worked for their crops. But when you have a look at these plots here, I've just put those alignment plots that I showed you earlier with those misaligned sites highlighted on red and orange. But I've just done a simple PCA. I've chucked all of those environmental covariates into a PCA to see whether they explain potentially why some of these sites are misaligned. And so let me just put that forward. I'll just put them all up. I guess, for example, scatter, you see this misaligned site has been spread apart away from the other data based on the patterns within the environmental covariates. But for Horsham, that pattern isn't really clear, and we've got, for example, 2023 and 2018 clustering with 2020, which is a site that is aligned. This was not the result that I wanted, and I thought I would have a really neat story, but obviously, this was just a preliminary look, but I also did more formal testing, which I don't have time to talk about today, but regressing these covariates, looking at the response against these covariates, so reaction norms, as well as fitting these covariates into FA model, and Daniel's got his paper from 2022 on the method on how to do that, and I found similar results, that these covariates weren't really explaining much of the genetic variance in the G by year. And even though that's not a great result for what I was really hoping to find, it really did provide some key thinkings and key steps moving forward, and key recommendations, I would say. Thank you. So I guess the first thing is G by E is complex, as we all know, but G by E in chickpea and legumes is even more complex. So legumes, all of them are quite indeterminate, and for those who aren't familiar with what indeterminacy is, it means that there's no real clear transition between vegetative and reproductive development, and often the plants will flower multiple times, or if it's flowering and then it's wet, then it will go back to being vegetative, and then, or if it's hot, then the plant will lose all of its flowers and produce more flowers again. So these plants have this innate adaptive response, so how can we really start teasing apart G by E with such a complicated physiological process? Environmental covariates add value, but many drivers remain uncaptured. So I've just taken just standard measurements from a weather station, but as I mentioned, we're working across a really big landscape. We need information like soil type. Potentially they will manage different. I know some sites just work on a flat bed, while others work on a raised bed. So how does that influence some of the results? Disease, that is data that I wish I could have, because I think that is, and I'll speak about it later, contributing potentially to some of the noise in the data. Exploring more targeted environmental covariates. It's been really challenging, and I think the invited speaker talk was really fantastic in highlighting this, was phenology is really key, and environmental covariates at certain stages of development really drive productivity. And so I often see, and unfortunately this is the case with the chickpea breeding program, is we don't capture any flowering or phenology data. And particularly when you're working on a crop that is incredibly indeterminate, it's really important to have this data. So if I could make any gentle recommendations, please capture any type of phenology data. Okay, lucky there's one more point. But potentially having more targeted covariates may allow us to maybe disentangle some of this G by E more specifically. And I know some crop modelers may say just use modeled flowering data. Well, I've tried, and unfortunately it doesn't work. And that's just because for chickpeas and many legumes, these crop growth models just aren't quite there yet. And when you're trying to look at diverse material, working on a flowering model that's based on one standard cultivar, you're not going to get really accurate results. And then last point that I was speaking to a colleague about, which I think is really interesting and it makes sense, everyone is trying to use breeding data now to pull apart these really big questions. So can we look at, like in my case, particular stresses within the breeding data? But if you've got environments that are really extreme in heat, but you've got all these sites in the middle that might be noisy due to disease or management, which is information that I don't know and isn't captured, then how can we make some of these conclusions? And should we be using breeding data to try and answer some of these questions, or do we need to actually start designing experiments that are a little bit more targeted? So those are my learnings from my unexpected results. But thank you so much.